button only because I'm afraid I'll forget, and I don't want to do this twice. All right. Oh, it looks like my connection is poor, so we'll do the best we can here. All right, everybody, don't forget your exam was released this morning at 8 a.m. It's due in 48 hours from the time it was released. So far, I've given 3 classes exams. Only one has had to turn their exams in, and sure enough, problems already. So hopefully I won't have to post on Blackboard announcements what I had to post for that class, because that class is actually my second semester biology majors. And yet I'm getting the most trouble with them. Apparently students think they can negotiate their grades and when they turn in things, and that's not it at all. But hopefully y'all will stay on the straight and narrow and take care of your responsibilities, okay? But make sure you get that exam done as quick as possible and submit it to me. Don't wait till the last second. I get some students saying that, well, I had internet connection problems. Well, the mistake that was made wasn't the internet connection. It was waiting till the last hour or minute or whatever, okay? So get on top of that. Okay, listen carefully. We're on the home stretch with the labs. These last 3 labs, including today, Cancer Gene Lab, they're usually the labs that most students tell me that they enjoyed. As a matter of fact, I've had students tell me, why the heck don't we start with these instead? So I want to come to class more. But all 3 labs, okay, at the center of all 3 labs is the compound DNA. Okay, so make sure you review chapter 3, and we will, okay, go over DNA a lot after this, starting on Thursday, chapter 10, 11, and 12. Okay, they do relate some to the labs that you're going to be assigned, and DNA is going to be at the heart of it, okay? So make sure you go over chapter 3, organic compounds, nucleic acids, and go over that background, and we'll do some of that in class on Thursday too. Okay, so today's lab is a cancer gene lab. It's a 2-part lab. I posted lab 10A and lab 10B. Lab 10A focuses on creating a cancer gene pedigree. If you remember, a pedigree is, I describe a pedigree as a genetic family tree, okay, where you track a single trait. So in lab 10A, you're going to be creating a pedigree, which I'll go over some of the symbols and stuff, giving you a comparison and an idea what you're going to do. And in that pedigree, okay, that you're putting together, lab 10A gives you information on a lady's family named, a lady named Valerie, gives you information on her family. And the trait you're tracking is cancer. Now the lab itself isn't just focused on cancer. It's focused on a type of cancer that falls under genetic disorders. It's called Li-Fraumeni syndrome. Okay, Li-Fraumeni syndrome falls under recessive genetic disorders, and we'll talk about the gene that's at the heart of this genetic disorder. Okay, Li-Fraumeni syndrome is not only a recessive disorder, okay, that falls under results in cancer, but the type of cancer that occurs from this type of genetic disorder, it could be pretty much any type of cancer anywhere in the body. Okay, so the way it's going to work for the pedigree, as I'll talk about more, is you're going to put together a pedigree, as I'll go over it, and then whoever had cancer, period, just had cancer, okay, then you're going to mark that down by filling in the shape, and we'll talk about that more. Then what you're going to be doing, lab 10B, is a cancer gene detection lab. Normally what you would do is you would come in, I would give you some DNA samples, I would go over what's in the samples, and then you would perform a technique called DNA gel electrophoresis. The gel is made up of a seaweed extract called agarose, which I'll show you in a bit, okay, and that gel, that agarose gel, okay, when we put it into, dissolve it into solution and allow it to solidify, we have a porous gel that allows us to separate DNA based on size, or if you want to call it length, okay? Now, what we're doing is what sometimes will occur for a person who has to see an oncologist because they've been diagnosed with. Okay, I think we lost each other. I stopped talking there. I'm trying to pay attention to this. So what will happen is they'll ask you for your family history. More and more, they're starting to come across different forms of cancer, may be, how do you say, inheritable, could be passed on from parent to offspring. But other forms of cancer are due to what we call a spontaneous mutation, and therefore they're not passed on. They weren't inherited. And so this is basically what you're doing. But what's going to finally determine whether Valerie's cancer it falls under an inherited type of genetic disorder called Li-Fraumeni, or whether it's a spontaneous mutation, therefore she doesn't have to be concerned about passing it on to her children, even though she does have to be concerned about her health, will be the DNA analysis, which falls under cancer gene detection. Okay, let me click the little hand now so I don't forget. All right, we're already having trouble and we just got started. System, but on my end, Blackboard's just throwing me all kinds of warning signals. So let's try to hang in there and do the best we can. Okay, so cancer and the cell cycle. Cancer involves uncontrolled cell growth, usually caused by a genetic mutation of a proto-oncogene or a tumor suppressor gene. Okay, well, we covered that in Chapter 8, Part 2. Okay, now, when cells become cancerous, okay, they start growing out of control. They start dividing out of control. And the type of cell division that they're going through is the cell cycle of mitosis. Something has happened to where either the proto-oncogene has been mutated. Boy, this thing is not cooperating. I'm actually trying to change the slide, everybody. Let's try it one more time. Okay, it might click twice. So what they found is that for many forms of cancer, it can be tied into one or two classes or groups of genes, either proto-oncogenes or tumor suppressor genes. Okay, we talked about proto-oncogenes before. They code for growth factors. Okay, and what can happen sometimes is those genes could undergo a mutation that results in cancer. Therefore, when the proto-oncogene becomes mutated, becoming a cancer-causing gene, they just now call it an oncogene. And don't forget, onco means cancer. Okay. But there's another class of genes, and it's actually this other class of genes that I've heard about the most being associated with cancer. It's tumor suppressor genes. These genes code for proteins that are responsible for regulating cell growth, in other words, cell division. Their job is to check for mistakes. If there's any mistakes, hey, I need to make sure I try to correct those mistakes so that the cell doesn't become cancerous. One of the most popular, and for the longest time, the most popular gene associated with cancers, different types of cancers, was a p53 gene. Sometimes it goes by the TP53, but you can know it as a p53 gene. The p53 gene, which we covered in lecture, okay, is a tumor suppressor gene. It contains genetic information that codes for a protein called p53 protein. Often they just say p53. p53 gene codes for a protein called p53. On this diagram down here, it's the diagram I used to use when I used to teach at another college, upper level, cell and molecular biology course. And we used to use this diagram and tell the students, look around the edges of the diagram. You can see p53 is mentioned very frequently as you go around. And wherever p53 is mentioned, it's always next to where it says DNA checkpoint. If you look at the inner circle here, okay, I don't know if y'all can still hear me, but that little hand's about to start flying around. I didn't realize it wasn't following me. But if you look at the inner circle there, you can tell just by the fact it says G1S and G2 phase, they're talking about interphase. So the M phase in this case represents the mitotic phase involving mitosis. So p53 is a protein that's, its job is to check for any damage to DNA. In other words, any mistakes or errors. And if there are any errors or damage to the DNA in particular, its job is to repair it. If the damage is too extensive or the error is too extensive, its job is to commit the cell to apoptosis, to prevent it from becoming a mutant cell, such as a cell that becomes a cancerous cell. So our body has built-in fail-safe mechanisms. Geneticists, okay, with the p53 gene, they found or discovered the p53 gene. The gene loci for the p53 gene is on chromosome pair 17 or on chromosome 17. So I'm showing you a karyotype where they circled it. When it comes to a genetic disorder called Li-Fraumeni syndrome, okay, that results in an offspring inheriting mutated p53 genes that result in a phenotype of the offspring being the development of cancer, in any type of cancer. Okay, geneticists, that further led geneticists to say, hey, you know, we need to figure out what's going on here, along with just cancers in general. Eventually, they figured out that the p53 protein was coded for by the p53 gene, and they discovered the gene loci was on chromosome 17. Well, if you recall, I have a karyotype there. The first 22 pairs of chromosomes are our autosomes. They have nothing to do with sex, okay, but they are all homologous, and this is something you need to remember, okay, because when I go over the DNA analysis part, I'm going to tell you to think of each DNA sample, with the exception of one. I'm going to tell you to think of the other 4 DNA samples that you're given as containing a homologous pair of chromosome 17s. So remember, they're homologous in terms of size, in terms of where the centromere is located and in terms of genetic information they carry. Well, the gene loci for the p53 gene is located on chromosome 17. The other thing I want you to note is notice where they're showing you the gene loci is. It's towards one end of the chromosome. This is important for you to know, okay? Because there's something that can be done to chromosome 17s that are carrying the mutated p53 gene that's been tied into causing Li-Fraumeni syndrome, okay? And that's something you're going to end up, that what I'm going to end up talking about actually results in anytime the p53 gene here at the end of chromosome 17 is actually mutated and is now a cancer-causing gene, we're going to talk about how geneticists discovered we can actually cut the DNA wherever this mutated p53 gene is located. Therefore, we can take one large piece of chromosome 17 and cut it into two smaller pieces. But the two smaller pieces are not going to be equal sizes. So please remember that. Notice where the location of it is. This is going really bad. This little hand is not following me. I'm sorry, everybody. Can y'all still hear me out there? Okay, because it's saying I'm having trouble and it says reloading the session. Okay, I'm going to keep going as quick as I can then. Yeah, it keeps cutting out. Hopefully the recording will come out better. I'm sorry, everybody. I'm surprised I'm having this much trouble in the morning, but I'm not surprised. Okay, let's move forward best we can, and this is being recorded. Li-Fraumeni syndrome. This is a genetic disorder associated with cancer, but Li-Fraumeni syndrome, if an offspring inherits this genetic disorder, again, they can develop any type of cancer. But the cancer, the cause of the cancer has to do with the mutation of the p53 gene located on chromosome 17. Now, Li-Fraumeni syndrome is a recessive genetic disorder. Therefore, the mutated p53 gene associated with the syndrome, it's considered a recessive allele. So we use a lowercase b to denote the mutated p53 gene that causes Li-Fraumeni syndrome. The normal p53 gene on chromosome 17, it's considered the dominant allele. Therefore, in order to inherit Li-Fraumeni syndrome, you have to be homozygous recessive, meaning you have to have inherited a chromosome 17 that's maternal and a chromosome 17 that's paternal, and both of them at the very end of the chromosome 17s at the same gene loci have to be carrying the mutated p53 gene, the recessive allele. If you are homozygous dominant or heterozygous dominant for this allele, the p53 gene, on your chromosome 17, your phenotype should be normal. But if you're heterozygous dominant, you are a carrier. Now, what they've discovered is that the mutated p53 gene, the one that is a recessive allele, they've discovered it contains what they call a hotspot. And what they mean by a hotspot is a DNA nucleotide sequence, okay, where the bases, it has a certain combination of bases. So when a geneticist sequences the p53 gene, if they come across a specific combination of bases in that p53 gene, they know that that's not only what's considered a hotspot for the p53 gene, but they know that means the p53 gene is the mutated one. Therefore, it's the recessive allele that causes Li-Fraumeni syndrome if you're homozygous recessive. So down here is what I mean by combination of bases, and we'll go over this more starting on Thursday, okay? The key to the genetic information stored in our DNA, the key to that genetic information is in the bases, okay? So what they found is within the mutated p53 gene, there is what they call a hotspot. And the way they explain this hotspot is when they sequence the DNA of the mutated p53 gene, there's a small stretch in the gene that reads like this. The bases are read as CA, GC, TG. Whenever they see this combination of bases that are part of larger structures called DNA nucleotides, whenever they see this combination of bases in this order, they know they are looking at a hotspot, and therefore they know they're looking at the mutated P53 gene, which is a recessive allele. Okay. Now, the P53 gene mutation, as I said, it can lead to different forms of cancer. So Li-Fraumeni syndrome, okay, is a recessive genetic disorder. Okay. It's caused by a mutated P53 gene, which functions as a recessive allele that results in cancer, but it can be any type of cancer. Now, this is important. Please listen carefully, because this is supposed to help you analyze your gel. Because what you're doing, the purpose of this lab is to try to figure out, is Valerie's cancer due to a spontaneous P53 mutation in her somatic breast cells, or is Valerie's cancer due to Li-Fraumeni syndrome, meaning that her breast cancer, okay, that cancer was inherited through mutated P53 genes. Therefore, this is the way it works. If the offspring has inherited a genetic disorder, such as Li-Fraumeni syndrome, then that begins at fertilization of the egg by the sperm. Okay? We talked about this. Sperm is haploid, carries one set of 23 chromosomes. The egg is haploid, carries one set of 23 chromosomes. When the sperm fertilizes the egg, they're not showing it here, but when the sperm fertilizes the egg, that fertilized egg will transform into a single cell, a single diploid cell that is known as a zygote. The zygote is diploid. It contains two sets of chromosomes. One came from dad, one came from mom. So down here on the bottom left, a zygote. It's a cell that contains a complete set of chromosomes, therefore it contains a complete set of genetic information. And those chromosomes, and therefore that genetic information, have been inherited from mom and dad. Now listen to this carefully. Every cell that makes up your body came from this zygote. So that's what I'm trying to say over here in this figure. The trillions of cells in your body all arose from one original cell. That one original cell is the zygote. So if it's a recessive genetic disorder, such as Li-Fraumeni syndrome, okay, then that means the offspring had to have inherited a chromosome 17 through dad's sperm and a chromosome 17 through mom's egg that were both carrying the recessive allele, the mutated P53 gene. Therefore, when the sperm was fertilized by when the egg was fertilized by the sperm and transformed into a zygote, that means this diploid cell, the zygote, okay, inherited or has in it a homologous pair of chromosome 17s that are both carrying mutated P53 genes. Okay, therefore homozygous recessive. Now, as this one zygote goes through several rounds of cell division involving mitosis, creating the trillions of cells that form your body, that means every single cell will also be carrying a homologous pair of chromosome 17s that both have the recessive allele. Therefore, if a person inherited a genetic disorder, it doesn't matter if you analyze the DNA in their cancer cells or if you analyze the DNA in their normal somatic cells, you will detect the recessive allele because it's in every cell of their body. We just don't know which cell will turn cancerous. If you inherit the genetic disorder, okay, that information will be carried in every cell of your body because every cell of your body came from the zygote and that was formed from the egg and sperm. However, if the cause of cancer is not due to an inherited genetic disorder, then that means when the sperm fertilized the egg, one or both of these was carrying a chromosome 17 that had the normal dominant allele, the normal P53 gene. Therefore, when that zygote divided into every cell that makes up the offspring's body, every cell inherited a homologous pair of chromosome 17s that were carrying the normal allele, the dominant allele. Therefore, when you analyze the person's cancer cells, Therefore, when you analyze the DNA in the person's cancer cells and compare it to DNA extracted from normal cells of their body, you will not get the same results. Okay? The results for the DNA analysis of the cancer cells will be different from the results of the DNA analysis from the normal cells. So this is how you're going to determine in the end, was Valerie's breast cancer due to an inherited disorder known as Li-Fraumeni syndrome, or was Valerie's cancer due to a spontaneous gene mutation in one of her somatic cells? Remember, somatic cells make up the majority of the cells of your body. They're all diploid. They all contain 23 pairs of homologous chromosomes, but the genetic information in those somatic cells is not passed on to offspring. So the cause of breast cancer has been associated with both inherited disorders and spontaneous mutations. So what you're going to do is the pedigree and a DNA analysis to determine, is Valerie's cancer due to an inherited genetic disorder called Li-Fraumeni syndrome, or is it due to a spontaneous mutation? Now listen carefully. When you create the pedigree, it's based on a lot of assumptions. However, when we perform the DNA analysis, that's what will let us know two things. Was the pedigree correct? Did it tell me correctly whether her cancer was inherited or spontaneous? And therefore, the DNA analysis will tell you if Valerie's cancer was inherited or spontaneous. Okay, so listen carefully. Lab 10A, you're going to create a pedigree. Okay, I've had some students tell me, well, I don't know how to create this. I don't know how to. Those of y'all that are here have been doing fine, but I've been having to let other students know, look, we're not in week 1, 2, or 3 of the semester. So create the pedigree. I don't mind if you create the pedigree by hand, take a picture of it, and then copy and paste it into your document, whether it's PDF or Word document, okay? So you can do it that way. That's the way I do things sometimes, especially with pedigrees, because it's a trial and error. This pedigree you're going to create using the information given in Lab 10A, you got to read through it carefully. Read through it carefully, because it's going to do my brothers, sisters, uncles, cousins, granddaughter, that kind of stuff. And then it's going to give you information on somebody that you think is a younger offspring, okay, in the pedigree you're creating. But you got to read through it a second time and realize, oh, wait a minute, they passed early on. They maybe lived to 1, 2 years of age, but they actually go higher up in the pedigree. What I mean by that is the oldest generation, when you create a pedigree, goes at the top. The youngest generation goes at the bottom. When you construct your pedigree, it should look similar to this. For some reason, I have students start bending the lines and what have not. That's not how it works. Now remember, when you create the pedigree, it tells you in the lab, square for male, circle for female. Pedigree is a genetic family tree tracking a trait. The trait you are tracking, in your case, will be cancer. Here, the example here is deafness, but you'll be tracking cancer. So listen carefully. Whoever just has cancer, period, you will fill it in, okay, to indicate, okay, they have the trait I'm tracking. They had cancer. Some of the individuals will be deceased. So if they had cancer and they're deceased, what you will do is draw a line through the shaded symbol. If they didn't have cancer and they're deceased, it doesn't matter if it's a square or circle, you will simply draw a line indicating that they are deceased. Now that's not on this pedigree, but I'm drawing it out here on the recording, okay? So again, squares represent males, circles represent females. If they have the trait you are tracking, which is cancer, color it in. If the person is deceased, if they are dead, they have passed away, doesn't matter if they had cancer or not, draw a diagonal line through it, okay? Now the way the pedigree is going to work, oldest generation goes first, okay? Oldest generation goes first. You can just follow this if you want, okay? So notice they put a square here for the male, a circle here for the female, and then you draw a line going directly left to right. Then you draw a line coming down the middle of that line to indicate these two individuals had children, okay? If they had more than one child, then what you will do is draw a straight line down far enough to leave yourself some space to add in some additional information I'll talk about. Then that line will branch out, okay? And the way you'll do it is you will list the offspring, oldest to the left, going down to youngest to the right. So once you get to that second generation, how do you say what symbol you use, circle versus square, for female versus male? It depends on who's the oldest to the youngest. So you can see this youngest generation here. They've just listed them. The way it works, oldest on the left, all the way down to youngest on the right. And if they have the trait, fill it in. Now you want to leave some spacing, because underneath the symbols, what I want you to do is put the genotype that you think they have. Okay, now if you notice here, a couple of these empty symbols, they still put them down as heterozygous dominant. Over here, notice they put a couple of empty symbols, but they didn't give you the full genotype. They only gave you one or two alleles. You may have to do this because you may not be sure if they're homozygous dominant or heterozygous dominant. However, you're going to be biased. Whenever the offspring has cancer, if you think there's a pattern being created to where it looks like a genetic disorder, then you can assume they're homozygous recessive, okay? Whether they're alive or dead. The other thing you're going to do is beneath the genotype, you're going to put down just their initials. So like Ray Earl is one of the family members. You don't have to write out Ray Earl, just put RE beneath the genotype. So again, beneath the symbol will be the genotype, and then beneath the genotype will be just the initials of the person. If they only give you one name for them, such as Omar, then just put an O. Okay, you don't have to write out the full name. You can if you want, but you don't have to. So you'll create that pedigree, okay, and that pedigree is going to let you know, hey, on paper, does it look like they might have inherited, that Valerie might have inherited the type of cancer she has? It may be due to Li-Fraumeni syndrome, which is a homozygous, or which is a recessive genetic disorder, meaning she would have to be homozygous recessive. Okay, so that's the pedigree. Any questions on the pedigree? So make sure you read through that pedigree one time, okay? And reading comprehension is very important, all right? Listen, I'm not looking for perfect on that pedigree, but I'm looking for damn good, okay? It's all right. I know it's not easy to do, especially online. Okay, but what's going to let you actually know whether Valerie has Li-Fraumeni syndrome or if her cancer is due to, her breast cancer is due to a spontaneous mutation in her somatic cells is lab 10B, where you actually perform a DNA analysis. Okay, so to determine if the cause of Valerie's breast cancer was inherited or spontaneous, you are actually going to analyze DNA samples from her normal cells and her cancerous cells. So if you look in your lab, I believe it's on page 2 or is it 3, there's a table. And they give you 5 samples, A through E, okay, and they tell you how you're going to load them in the gel. They say Wells. And then for each sample, it tells you what's in each sample. So, and I'll go over this some when we get to it. But for now, what geneticists discovered is that that hot spot found in the mutated P53 gene, okay, that's associated with causing Li-Fraumeni syndrome, geneticists found that there are proteins called restriction enzymes that will cut that hotspot in a mutated P53 gene. In other words, there's a combination of bases that are recognized by a protein enzyme, and that enzyme will go up to the DNA, bind to the DNA like it's showing you here. Okay, if it finds that hotspot in the mutated P53 gene, this protein enzyme called a restriction enzyme will bind to the DNA where that combination of bases are, and it'll act like a pair of scissors. It'll cut the DNA. Okay. Therefore, the way we're able to take advantage of this is, as I told you before, the P53 gene, its gene loci is not only located on chromosome 17, but it's located towards one end of chromosome 17. So what you're going to end up, what you would have ended up doing is being given DNA samples that contain a homologous pair of chromosome 17s in solution, along with the restriction enzyme that recognizes this hotspot in the mutated P53 gene or in the recessive allele. And you would have also been given a sample of DNA that contained a homologous pair of chromosome 17s that were both carrying the normal p53 gene or the dominant alleles, so that you could compare what the normal or what the dominant alleles would look, how they affect the shape or size of the chromosome 17 compared to if the recessive allele is located when you combine it with the restriction enzyme. And that did not come out as good as when I tried to rehearse this. I'll show you something else in a bit. The way you're going to analyze her gel, though, is not only by exposing a homologous pair of chromosome 17s isolated from different cells of her body to a restriction enzyme that will cut the recessive allele, but by taking those DNA samples that contain the homologous pair of chromosome 17s and have been exposed to the enzyme, you will take those DNA samples in solution and you will perform a technique called DNA gel electrophoresis. Okay, DNA gel electrophoresis. The gel is in reference to, we use a porous agarose gel. Okay, agarose is a seaweed extract, comes in a powdered form. You dissolve it at a high temp into solution in a buffer solution, and then you let it cool and it'll solidify into a gel that has tiny pores. That's the gel part. The electrophoresis is that researchers found you can apply an electrical current to this gel and the current will run through the gel. Okay. Now, why am I talking about this? Well, biochemists discovered DNA is naturally negatively charged, and we'll go over this on Thursday. DNA is naturally negatively charged. So geneticists realized, you know what? Taking advantage of agarose gel, taking advantage, okay, of electricity, and taking advantage of the fact that DNA is negatively charged, we can actually use this agarose gel along with electrophoresis, the electrical current, to separate DNA based on size. Because the agarose gel is porous and DNA is negatively charged, what you would have done is you would have added your DNA into what we call wells on one side of the gel. Okay, and I'm going to go over this a little bit more in detail. And then to that side you added your DNA, you would have had a negative charged electrical current being emitted because DNA is negatively charged, and because we're having a negative charge emitted here, that causes the negative charges to repel each other, or more to the point, that pushes DNA into the gel. From the opposite side of the gel, a positive electrical current is emitted, which attracts the DNA or pulls it through the gel. Therefore, between the push and pulling that's going on, this allows DNA to migrate through the gel. Some of the fragments of DNA are very large, so they have a hard time getting through the pores in the gel, so they move very slow. Sometimes the DNA fragments are very short or small, therefore they're able to move through the pores much quicker. Therefore, when you look at your DNA gel, okay, if there's a large piece of DNA present, it'll be located towards the top of the gel, close to where you added your DNA sample to begin with. And if there's a small piece of DNA, it'll be located more towards the bottom. Now, this is important because remember what I said. Chromosome 17 is where you will find the gene loci for the p53 gene. And that gene loci is located towards one end of the chromosome. So if the enzyme finds the recessive allele and cuts the mutated p53 gene, we end up with two smaller pieces of chromosome 17, but one of them still larger than the other. And I'll show you in a sec here. Okay, let me speed this up a little bit. Okay, so the agarose gel normally, okay, it's a slab that's laid inside of a gel box, and it's actually lying parallel to the bench top you would have been working on. And then we cover it with a buffer solution. The purpose of the buffer solution, it has salts to help conduct the electrical current. And then on one side of the gel would have been wells. So on that table I gave you that lists the samples, okay, and where the DNA came from in those samples, it talks about well 1, well 2, well 3, well 4. You would have loaded your DNA samples in the gel in wells located closest to where the negative charge would be emitted, so that the DNA would be pushed into the gel, and then from the opposite end, the positive charge would have been emitted and would have pulled it through. The thing is this, okay, we don't want your DNA sample to float away when you add it to the well because the gel is covered in a buffer. So your DNA samples would have been combined with a loading buffer that contains two key components. One is glycerol. Glycerol is a viscous solution that makes your overall DNA sample very dense. And if you look down here, you can see the wells are filled with DNA samples that are in loading buffer. The glycerol is making sure the DNA sample is heavier than the surrounding buffer. Therefore, notice how nicely the DNA samples settle into the well rather than floating away. However, we can see them. Well, the reason why we can see them has to do with the tracking dye. There's also tracking dye in this loading buffer so that you can see the DNA that you're loading into the well. So what would have happened is you would have taken a micropipette with a micropipette tip. You would have put the tip into the buffer solution. You have to have it into beneath the surface of the buffer solution, but just above the well, okay? We don't want to load it above the buffer solution because it'll just splatter all over the place, but at the same time, we don't want to stab the gel to death. You can see the person here is loading it perfectly. Then the tracking dye allows you to monitor the progress of your gel electrophoresis, okay? The tracking dye is made up of very small negatively charged molecules that will move faster than your DNA. That way you know when the tracking dye gets close to the bottom, your DNA is somewhere just above it, you know when to stop the process, cut off the electrical current. If you had done a perfect job and not missed any of the wells that you added your DNA to, your DNA gel would have come out looking like this. Okay, I'm only going to go over a couple of these. The rest are up to you. But listen, in the table in Lab 10B, it tells you sample A, which goes into well 1. So this is well 1. We count the wells from your left to your right. Well 1, 2, 3, 4, and 5. This is all in Lab 10B in the table. It tells you in that table also, well 6 will be empty. So here's lane 1 that contains sample A. Sample A is what we call a DNA standard. It comes with companies. What they will do is they will synthesize different sized molecules of DNA. In other words, different sized fragments of DNA. They will combine them into a solution and sell them. Okay, and what we do is what you would have done is you would have loaded sample A here, and after electrophoresis was finished, you would have noticed, hey, look, this is telling me that I had multiple fragments of DNA of different sizes. Some were very large, some were small, some were very small. Now listen carefully. Each of these bands represents several pieces of DNA that are the same length. So this one up here, this band that's at the highest point of the gel closest to the well, it consists of several molecules of large fragments of DNA. But since all these molecules of DNA are the same length, the same size, we see one band. But there's actually many, many molecules of DNA there, the same fragment, same length. This is sample B, lane 2. Okay, this is your control that you will use to help compare to these other 3 samples, which come from Valerie's tissues. One of these 3 samples loaded in wells 3 through 5, which are samples C, D, and E, all come from Valerie's tissues, but only one of the 3 comes from Valerie's breast cancer cells. Okay, but you want to start with lane 2, sample B. Sample B contains a homologous pair of chromosome 17s that are both carrying the dominant allele. So I drew it out here. Okay, imagine these look exactly the same. The little circle represents a centromere. So we had, and I put down here, control, sample B. A homologous pair of chromosome 17s, and both of them are carrying the normal P53 gene, so I put an uppercase B. Over here is my ugly gel with the well. And I said for each of those DNA samples, B, C, D, and E, imagine they have 2 chromosome 17s. In other words, imagine you have a homologous pair of chromosome 17s in samples B, C, D, and E. Therefore, sample B, which was given to you, okay, which are controlled, sample B had a homologous pair of chromosome 17s that were both carrying the dominant allele. Therefore, neither one contained a hotspot, this sequence. Therefore, the restriction enzyme doesn't cut the DNA that makes this homologous pair. Therefore, this homologous pair stays the same size, the same length. So in a gel, you don't see these two separate. Both of these are the same size. So in your gel, they're going to move at the same time together as one. So they appear as one band. But what you can't see is that one band, which you can see over here. That one band actually has two molecules of DNA that form your homologous pairs of chromosome 17s. But they're both the same size, so they're moving together. Now, I'm not going to go over the other three because that's your job to go over that there. Yes, these labs require some thinking. So listen, one lab report, combining labs 10 and A. Create your pedigree, give me a cover page, create your pedigree for 10A. Just title it pedigree. Okay, like I said, you can do it by hand, take a picture, copy and paste it into the document, and then answer the questions, okay, for on the, what you would call it, I'm sorry, label your gel pick on page 2. Okay, so give me a copy of that gel pick on page 2, and then answer the questions on the last page. And listen, I gave you the gel pick 2 times. It's in the PowerPoint and it's in the Word doc in the lab that I gave you, Lab 10B. Okay, so you have it 2 times, that gel. So make sure you label it. And how you're going to label it? Well, it says in your Lab 10B Word doc, use the information from the table to label it. Okay, the usual bit. Let's do a week from today, by or before 9:30 a.m. Are there any questions? It's a good lab to think through, everybody. It is a good lab. Take care of your exam. And then, like I said, we'll cover more on DNA on Thursday, but definitely get your exam out of the way first and then take care of your lab, okay? All right, if there aren't any questions, I'm going to stop the recording.